Hey everybody. So for exercise 3, I thought I'd put a video together to kind of walk us through a little bit of functional morphology, which is the lab exercise for this week. Now, this particular lab exercise, I've chosen bivalves, bivalve mollusks, as an example that we're going to use for our functional morphology exercise. The goals of today are to look at the morphology of different types of bivalve mollusks and learn a little bit about the role of shape into the function and how they interact with their environment that way. And along the way, we're also going to review some very basic bivalve anatomy and some common life modes of some of these different groups, so we have a context to put them in. And this will then help you as you go through the online exercise looking at functional morphology in bivalves. So we'll start with the image on the left, which is the dorsal anatomy of your basic bivalve. In this case, we're using a scallop or the genus Pecten. Up at the very top, we have the dorsal side. This is the hinge line. So that's that slightly curving but more or less straight line there at the top is where the two valves or the two shells of our bivalve would be attached. Now, coming off of either side of this particular taxon are broad auricles, or these flanges that stick out. And this actually aids this particular group of bivalves in attaching to the seafloor. This is not a burrowing in bivalve. They actually will attach with what's called bisial threads. Some of them actually don't use the bisial threads, and they actually swim. So bivalves are actually a little bit more of a diverse group than we give them credit for. And so just keep an eye when you're looking at anterior, posterior sides, and the ventral side. There is actually a front, back, top, and bottom to a bivalve. Now, when we look at the internal anatomy of the shell, not the soft tissue organism, but the shell itself, and that's the image on the right, this is looking more at a clam, a different type of bivalve, but some of the same features are present. We have that hinge line, you can see. Those two little elliptical structures next to where the arrow is pointing for the hinge line, those are the teeth, not for eating, but for attaching these two valves together. Some bivalves have these and some don't. The kind of swollen area that's kind of along the back dorsal surface of the bivalve is called the umbo. So when you're holding a shell, this is the part that kind of the bump that sticks up. There's an attachment surface for ligaments in this particular group, not really as much or as strong in some other bivalves like pectens. But I want to focus your attention to the inside of the shell. We have these muscle scars, the adductor muscle scars, a posterior and an anterior. Some bivalves have one of these, some have two. In both cases, though, they are adductor muscles. The function of these muscles is to keep the shell closed. When they relax these muscles, the ligament that's attaching the two shells together acts as a spring and opens them up. So when it comes to a bivalve mollusk, they can only close their shells actively. They passively open their shells by just relaxing those muscles. And the last feature I want to point out here is the pallial sinus. This kind of curvature inward towards the inside of the shell. And on the images of shells or any bivalve shells you look at, you'll actually see this. It's a line that forms around the internal margin of the shell, and that's actually showing you where the bivalve organism, the soft tissue organism, attached itself to the inside of its shell. Now, the pallial sinus is the location of that animal's siphon. It's a tube that bivalves use, some bivalves use, to extend outward. So when they're buried in the sediment, they'll stick this tube up through the sediment out into the water so they can breathe and feed. So the deeper the pallial sinus, the larger the siphon. So think of it kind of like a snorkel, but they're still underwater. So these are just some of the basic anatomical features that are going to be useful and you'll want to review back to as you're going through the exercise. Now we can take a look at bivalve body plans and modes of life. You're seeing the wide diversity of the different shapes and sizes bivalves can take and how they live. So for example, we'll just kind of go through some of the letters here. Figure A, you're actually seeing oysters. Oysters grow in, most oysters grow in these beds where they will attach these bisial threads to each other and they kind of secrete onto each other. They form one larger reef-like structure. So they're epifaunal, they're not infaunal. B and C are different types of scallops. Notice B is free swimming. That's because some species of scallops actually do this. They pull water in and out of their mantle so they can squirt through the water. And then figure C is showing you a different type of scallop that actually attaches with bisial threads, as does specimen D, which is a different type of bivalve, not a scallop. E, F, G, H, I, and the rest of them are all infaunal bivalves, and you can see the various forms that the siphons take. Now, figure I, that kind of weird triangular shaped, kind of spiky bivalve, that does a little bit of both. It's infaunal, it has the bisial threads, but it doesn't use a siphon because half of it's sticking out of the water. So I want you to pay attention a bit to the shape of these bivalves, of their shells, and what kind of environment they're living in. Notice that the really deep digging bivalves are usually elongate, but some are not. But most of them are. And that allows us to then make some predictions about fossil organisms that we find. Oysters, for example, can have quite a bit of variety. On the left, you're seeing a bed of modern-day extant oysters. Notice they have barnacles growing all over them, but they're all attached to each other. So there's kind of a strength in numbers to build a larger structure. On the right, however, we have a fossil of Exogyra, which is a Cretaceous oyster that's about the size of your hand. They're also about 4 centimeters thick. They're incredibly thick with their calcium carbonate shells, and they don't appear to have lived in beds like modern-day oysters. They were definitely epifaunal. They weren't free swimming or anything. They were much too heavy, but they have a very distinct shape. They're still a type of oyster, but they have a distinct shape that indicates that they're not forming in these clusters. Now, they may have been living in close proximity, because usually where you find one, you find many, but they're not attaching to each other. They're heavy enough where they don't need to do that. So again, the shape is giving us some hints about the lifestyle. Scallops are unique among bivalves in the fact that they're the closest thing to being truly symmetrical, like a brachiopod, but they're actually not. If you look carefully at the auricles on the anterior and posterior sides, or the left and right sides, you actually notice that they're not quite symmetrical. They're close, but not quite. But again, these will either be attached or nectic, or they're free swimming. Most of them are attached, but they're not really infaunal. You don't have to dig deep to get scallops. It's not like things like clams. You just have to dig for clams, not really for scallops. But again, we have basally attached scallops, and we have our free swimming scallops. So how can you tell which one is which by just looking at a fossil? Well, when we look at our modern day bivalve communities, we notice that there are some relationships between the shape of the shell and the lifestyle. So on our chart up at the top, you see all the white circles represent what we call umbonal angles, so kind of how narrow or wide the shell is. And then your x-axis, the x and y-axis here, you've got the umbonal angle and then the auricle asymmetry. So how asymmetrical are they? And so we add these two factors together and notice that there's a very clear distinction within Pectininidae, this particular group of scallops, for the ones that are basally attached and free swimming. It appears that higher umbonal angles and lower auricle asymmetry ratios will provide us with scallops that are more in the free swimming area. Meanwhile, our basally attached make up a greater portion of the ecospace, but they actually have very distinct characteristics. Notice that the auricle asymmetry is much higher than what we see predominantly with our free swimming. So there is a relationship here between the shape of the shell and the lifestyle of the organism. Mussels are another one of our favorite tasty bivalves. We'll look at a couple of different genera here. Mytilus and Modiolus are two common types of modern-day mussels, and they also have very distinct lifestyles. Modiolus is infaunal, but still using bisial threads, and Mytilus is epifaunal, using bisial threads. And notice, when you look at measurements of the two shell shapes, that Modiolus is elongate. It's more cylindrical shape than Mytilus. So a more cylindrical shape does seem to correlate, whether it's in mussels or clams or other things, with an infaunal lifestyle versus a more compact form being more adapted to an epifaunal, attached lifestyle. There's actually quite a lot of evolutionary convergence in bivalves, in mollusks in general. Here we see two totally different, distinct lineage bivalves, but notice both of them have the elongate shape. Does anybody want to guess what type of lifestyle these two bivalves have? I obviously can't hear you. They are infaunal, but they both evolved the characteristics that we would call infaunal bivalve characteristics independently of each other. Ornamentation in bivalves is more than just a way for us to identify the different kinds of shells. These actually serve a function. So on various type of bivalves, you'll see curves and crenulations and wrinkles and sometimes what looks almost like spikes or thorns coming out of them. These sometimes are a function of protection, but in many cases, they're hydrodynamic. These are characteristics that are going to help that organism not get washed away during a storm event or help it grip into the sediment if it's somewhat infaunal. So with that, you should be ready to start tackling exercise 3, which is on the functional morphology of bivalves. So this is a self-contained lab exercise that's all on Canvas, but I am making myself available if you have questions, and I encourage that. This is something that you are kind of meant to struggle with, maybe not struggle, but wrestle with a little bit because you're looking at modern day information about bivalves and trying to apply it to the fossil record. So you're making those educated assumptions and inferences. And sometimes that can lead you down some weird roads. So feel free to contact me with any questions. So work through exercise 3 on Canvas. This is going to be due Thursday, October 8th, if not earlier. Otherwise, I look forward to hearing from you, and good luck.